what we're going to do is that out of those 2 million nephrons, okay, I picked out 1 nephrons, okay, and I drew it down here, okay, and I also labeled it, and we did it together, right? So this one is afferent arterioles and glomerular capillaries, okay, and the afferent arterioles, okay. The afferent arterioles, when it breaks down, because remember, okay, before we go there, okay, one thing we have to talk about is that, as I said, nephrons are the functional unit of the kidney, okay. Nephrons play a very, very important role, okay, because they are very, very important, and we'll talk about that. So we have to go in very detail about this, okay. Now, one other thing is that if you come back here, okay, this afferent arterioles, when it gets breakdown, okay, it can make from like 20 to like 40 loops of this tuft. They call this tuft or network of capillaries, okay. And then this whole idea are called glomerular capillaries, okay. They are called Glomerulus capillaries, okay. That tufted network of capillaries, okay. They mix all this loop around, okay. And then that will go becoming efferent arterioles, okay. That's what we talked about. And then if I come back here, right here, okay, this guys, you see this I draw this in blue color. These are called mesenterial cells, okay. They're usually they're different. They're mesenterium. Okay, there are intramesenterial cells and there are also extramesenterial cells, okay. The reason intra means they're inside this blood vessels or blood inside around this, because that's what they call intramesenterial. Extramesenterials are usually, they're associated with the juxtaglomeruloparas, okay. They're outside of this capillary network. That's what they call extra, okay. And this mesenterials has some of the function too. Okay, let me just write down some of the functions of this, One of the functions of these mesenterial cells is that they have a phagocytotic functions, okay. They also have some kind of immunoglobulin functions, okay. They're very, very important for secreting like IgA, okay. And then they have a phagocytic functions, as I say, okay. They also have functions like, they can secrete like some kind of substance called lysozyme enzymes, okay. They have phagocytic functions. They have some immunoglobulin functions too, okay. And then phagocytic functions, they also can produce IgA immunoglobulins, okay, to fight for sort of like infections or sort of that kind of stuff, okay. That's just what I just wanted to quickly mention about that too. So basically, they prevents like any sort of like damages to happen in the glomerulus, the glomerular capillaries, okay. Now, we'll have to talk about what is the role of glomerular capillaries, but before we even get there, okay. You see the black color I draw? I basically draw that as a basement membrane, okay? Because every epithelial cells, endothelial capillaries, contains basement membrane, right? And these are called glomerular basement membrane, okay? And we'll talk about it in very detail about that. I did that in black color. It's very dense, but it's not very dense because they have 3 different layers. We'll talk about that too. And then these is C-shaped copper right here. These are called the Bowman capsules, okay? The Bowman capsules, and this right here is called space, okay. So these Bowman capsules are made out of 2 layers of cells, okay. One is this is inner layer cells. We call them an inner visceral layer of the cells, okay, and they have specialized type of cells. And this, this being called specialized type of cells, you have to remember this, please. It's called podocytes, okay. These podocytes, and we'll talk about it in more detail in here, okay. These are called podocytes, okay. And this makes the inner visceral layer of your Bowman capsules, okay? And right here, this guy right here, down here, this is the parietal layer, okay? And this is just made out of simple squamous epithelium, okay? This outer parietal layer of your Bowman space, okay? And then after that, what happens to that, it will make down into proximal convolutive cells, okay? And then proximal convolutive cells has those cuboidal brustborder cells, or so as like about 60 to 65% of reabsorptions happen from there. And we'll talk about that when we talk about the tubular fluid, okay? But right now, we're going to focus on this one, these structures. We're going to talk about this glomerulus and basement membranes and also the podocytes relationship, okay? So now, hold that thought, and then what are we going to do is we're going to use this and we're going to draw it here and we're going to go in very detail, okay? Now, what's this? And we'll talk about this, okay? Now, let me erase this, okay? Okay, guys, this is very, very important that you have to know, okay? So let's make this efferent arterioles right here, okay? So I'm gonna draw right here. I'm gonna draw my capillaries. Just gonna draw capillaries here, okay? So I'm gonna draw. Let's make this as a capillaries, okay? I'm making a endothelial capillaries, okay? Right here. So basically, drawing of blood vessels. Okay. Okay. Look. So let's make this as a efferent arterioles. So this is efferent arterioles, bringing blood to the kidney. Okay. And also this is my efferent arterioles. Okay. Efferent arterioles. Okay. Now you see I draw here. What are these? These are my capillaries. Okay. What kind of capillaries are this? These are my endothelial capillaries. Simple squamous endothelial capillary. These are lining these glomerular capillaries. These are very, very important, okay? The reason why is, okay, we'll actually make this a little differently, okay? I draw this a little differently, but we'll just make it this way right here. Let's make this as this. It usually looks like this because it's lining these capillaries, okay? And they have obvious nucleus, okay? Now, these capillaries are very, very important. The reason why these capillaries are very, very important is that these guys do have, these are intercellular, you see this, these are called intercellular cleft, okay? And they do have intercellular cleft too. Oftentimes we don't talk about this, but it's important. They do have intercellular cleft. But what is the unique of this capillaries, which is lining the glomerulus is that in the cells right here, in the endothelial, within the endothelial cell itself, they have like bunch of holes, okay? The bunch of holes, like I'm just gonna cut this like this here. Bunch of holes like this. And this space, right, you see bunch of holes right here? These are called, because a lot of pores or holes in it, we call them as a fenestrated, okay? What do we call this? This is called fenestrated. Fenestrated capillaries, okay? That's why these are called fenestrated capillaries, okay? And usually this is, if you want to remember the diameter sizes, it's about like 70 to like 90 nanometers of sizes. So anything particles, okay, that is smaller than 70 to 90 nanometers, that can actually easily pass through this, okay? That is something that you have to remember. Now, okay, that's why they're highly porous, okay? These guys are. And they do have intercellular cleft too. Intercellular cleft means the endothelial cells are joining together, right? Joining together, okay? And usually your endothelial cells, where do you see that? Usually you see this type of, without a pores, okay? Usually this intercellular cleft, usually you see that in a what kind of capillary? In a continuous capillaries, okay? Like for example, in the capillaries in your lungs, okay? And then what is covering this inner, then after that, these guys are sitting in the what? This is my glomerular capillaries, okay? We'll just write down glomerular capillaries, okay? And they're sitting in this guy right here. And what is this guy right here? I'm not drawing little bit, this way right here. This is called basement membrane. So we call them a glomerular basement membrane, okay? And we have to talk about this in a little bit in detail, because what happens, what is the unique properties of glomerular basement membrane is that it has 3 layers, okay? One is very, very dense right here. This is a middle layer, okay? So very, very dense. And it is usually this middle layers of this glomerular basement membranes are very, very dense, okay? Because it's very, very dense in electron microscope, we call them as a lamina densa, okay? We call them a lamina densa, okay? And then maybe this guy right here, it is maybe like, this is kind of like attaching with the inner layer of the endothelial cells. So we call them as lamina, okay? Lamina rara interna, okay? And then, okay, this is basement. This is this side, okay, it's not very dense. It's lamina rara interna because it's lining with the endothelial cells of the capillaries, glomerular capillaries. And here, there's another cells, okay? We have to talk about this. Let me draw this in this color right here, which is what is this? This is going to be my Okay, actually, let's not draw that. Let me just draw in a different way. We'll make this as these are going to be my podocytes. Okay, these are going to be my podocytes, and then we'll say these are podocytes food processes are. These are going to, let's make these food processes. These are podocytes food processes. Okay, so these are these podocytes food processes are lining on the basal membrane too. So these are called, this side we call them is a, okay, it's a middle layer, we call them a lamina densa because it's very dense. This is which is lining the endothelial cells of the cap, which we call them a lamina rara interna. This is, we call them as a lamina rara externa. Okay, this is what we call them. Okay, now in order for the filtration to occur, because remember, kidney extract out the urine, right? It has to go through all these different things like, look, endothelial cells, okay, then basal membrane, okay, and also this guy, these podocytes, okay, they all are sort of like creating a barrier for any things to actually filter out, okay, because there's a lot of these three, they have to go through the barrier to get to here, okay, to get to, let's say, this place, okay, like right here. If they pass this up to here, then we call them as ultrafiltrate, okay? That's what we call, that's where the word comes, the ultrafiltrate, okay? Because it's very unique properties of the kidney, okay? Now we have to talk about in very detail about what these are. So now, as I said, look, guys, in the endothelial cells, okay, what happens is that these endothelials which are lining the glomerular capillaries, they have these pores, okay? These pores are called fenestrated, okay? But remember, guys, but how do I know? But if I make this, okay, quickly, continue, like this is my, let's say, this is going to be my, okay, this one right here, right? This is my continuous capillaries, because anything that has to pass, it has to pass it from this area, right? From the intercellular cleft, okay? But if I make, okay, let's just, no, you don't need to make that. But remember, if I do this, right? But if I look at the electronic microscopic, right? How do I know whether it's a continuous capillaries or whether it's fenestrated capillaries? Okay, because remember, here around there, intercellular cleft, you might see a little bit of gap in there too, but that's why you may get confused, okay, whether you can able to tell whether this guy whether it's a continuous capillaries or fenestrated capillaries, okay, because how come you don't know that this could be, you may confuse, and you could say that this is not a fenestrated, but it is a continuous capillaries. You could think that this is not this, you could think that this is intercellular cleft. You could think that way, right? You could just think that way. You could just think like, oh, this is probably, maybe this is the fenestrated, but you might think that, oh, this intercellular, you might think this is intercellular cleft. Okay. So how do you distinguish the continuous intercellular cleft versus whether it's a fenestrated, okay? Well, one of the things that is so unique about this thing is that they have something called this thing, okay? What is this thing is, okay, let me just make it right here. Okay, they have this sort of like a slimy or something around here, okay? Coming like this. Okay? And what am I drawing this? Okay, you get to see that. Okay, but you don't see that in the intercellular cleft. What is that? That's called, okay, what is it called? Do you know this guys? It's called Glycocalyx, okay? Glycocalyx. So, in kidneys, you see this Glycocalyx, okay? So, if you look at the electron microscope, and if you see that in the pores, if you see this little bit of the Glycocalyx there, then you could able to say that is your fenestrated capillaries. Because what happens is that in the intercellular right here, like I made it here, you don't see the Glycocalyx there, okay? So, in order to distinguish whether it's a continuous capillary versus fenestrated capillaries, looking for the Glycocalyx. And this space right here, this one, we call them as a diaphragm. Okay? I don't know why, but they call this a diaphragm, if you want to remember that. Okay? Now, and this Glycocalyx also plays sort of like a negatively charged molecules, okay? Like, they can repel certain proteins, okay? Because they're negatively charged, okay? It also helps with the prevention of that. All right? Now, anyways, then after that, this endothelial cells or this membranes of the lamina interna, okay, they actually have certain proteins, okay, and that proteins have negatively charged proteins. And what are those proteins called? Which are called HEP, heparan sulfate. And heparan sulfates are negatively charged molecules. Even in here, in a lamina densa, raw externa, there's also contains of this heparan sulfate, and heparan sulfates are negatively charged molecules, okay? They have this also other types of thing called proteoglycans, like, for example, if you remember, it's called agrin, okay? Proteoglycan, heparan sulfate, these compounds called agrin, these are proteoglycans, okay? They have these base members are full of fibronectins, okay? Fibronectins, okay? And one more thing is that on all these guys, altogether, whenever we talk about the sugars and proteins, okay, because they are actually providing the negative charge for things, the protein, so that way protein does not excrete out from the kidney, because we need protein in our body, right? So we all together combined, they call them as a siloglycoprotein, okay? They all call these siloglycoproteins. And these siloglycoproteins are mixtures like sugars and all that kind of stuff, sugars and proteins. So they can actually help with the repelling the negatively charged particles, because these are negatively charged, okay? So anyways, basement membranes is full of negative charges, okay, because they have this fibronectin, this agrin, this heparan sulfate, okay, laminin, okay, they also have a laminin. And laminin number 11, if you remember this, okay? And proteoglycans, I'll just write down this. Proteoglycans, okay, like agrin, all right? And they have a heparan sulfate, you have to remember this, okay? They also have fibronectins, all right? The base members, all that stuff. So all that stuff provide negative charge, okay? And guys, we have forgot one more thing. There's one more thing, this actually usually surrounded on this one, on the lamina densa. There's very, very important compound there. What is that? That is called collagen. Which collagen? Number 4. Collagen number 4, okay? And these collagen number 4 also plays a very, very important role, because collagens are made out of what? The triple helix, right? The triple helix of the amino acids, right? And then what happens is that these guys also helps with it also plays a role of barrier, okay? Because collagens are negatively charged, okay? And guys, you should remember this, because there are a lot of conditions, a pathologic condition related to this glomerular basement membrane. Because remember, if somehow there's a mutation in the collagen 4, okay? Or then there is going to be a condition called glomerulonephritis, okay? Some of the kidney diseases condition that could happen because of that, okay? So it's really important that you remember this. There's in collagen, there's a triple helix, right? They have this on the at the end, there is an area where you can also form like antibody against it, okay? And that can give you some kind of global glomerulonephritis. We'll talk about when we talk about pathology of these conditions, okay? So now, quickly, here on the endothelial cells, they're about 70 to 90 nanometers. We talk about the Glycocalyx, okay? All that kind of stuff. So usually anything that is coming out, like larger cells, like red blood cells, white blood cells, they would not be able to filter out, okay? And also, the endothelium itself has this the Glycocalyx, and also because of that, it also repels some of the proteins, like larger proteins cannot filter out. But some reasons, let's say it did get filtered out some of the portions, okay? Then what happens is that any negatively charged have very difficulty to pass through this because of the basement membranes. Basement membranes are full of electronegative, and those electronegatives are provided by all different types of these proteoglycans, like agrin or heparan sulfate or even collagen 4, all right? Laminin number 11, all right? And then fibronectins, collagen 4, as I said, they are full of the they're full of electronegative. And what happens whenever the negative charge comes in, negative charge proteins, what happens, it will repel out, okay? So it doesn't allow to filter out, okay? That's what this glomerular basement membrane does, okay? Now we have to talk about this guy, and what are these guys are? Oh, let me quickly erase this, okay? So these guys are called podocytes, which are making the visceral layer of your Bowman capsules, okay? And these podocytes podocytes kind of look like an octopus, okay? You know, if you want to remember octopus, it kind of looks like that. It has like those finger-like projections coming out, okay? So if I could draw this right here, okay? Let's make this as this podocytes right here, okay? And it has a finger-like projection coming out like this, okay? These podocytes are also called foot cells, okay? They're also called, what do you call this? They're called foot cells, okay? And they have this protoplasmic extension. What do we call them as? We call them as protoplasmic extensions are coming out, okay? These protoplasmic extensions are called, we can call them end-fit, okay? Or they call them as pedicels, okay? They call it pedicels, okay? This end-fit, this protoplasmic extension that are coming out, we call them foot process or finger-like projection that's coming out. These are protoplasmic extension of these podocyte cells, and these cells are also called end-fit or pedicels, okay? And they're coming from everywhere, okay? So if I make this as, let's say this is the glomerular capillaries, right? The protoplasm, these are my finger-like projection. It's coming and covering like this, okay? It's covering and holding like this. So this is my finger-like projection. That's what is happening with this guy right here, okay? And one of the key aspect of this is, let's say, okay, I erase this, okay? Let's say another cells, if I make this another, like this. You see what I mean? Like right here? You see like right here? So this finger-like projection, and these are pedicels, okay? These are pedicels. The protoplasmic action, these are pedicels. When the pedicels come together, like, you know, from everywhere, it's also coming from all the way, it's holding together, right? So when they're coming together, holding, what happens? It creates a gap, okay? So it creates a gap. Okay, it creates like a space or gap. Like for example, like right here, you see this space right here, it's coming here, right here? This space, okay, let's just make a different color. This space created by these two pedicels, okay? They're created by these pedicels. This space is called so-called filtration slits, okay? The spaces like of these two area, we call them as I'll just make this as so these are two coming together. We call them a filtration slits. Okay, and usually that's like 40 nanometer in size. Okay, see how the filtration slits are created? So fluoroplasm extension, when they're coming together, there's a gap forming the space, okay? Those spaces by two pedicels, okay, when they're coming together, that creates something called filtration slits. But guys, don't get confused filtration slits with filtration slits diaphragm. Well, that's another thing. Okay, we'll talk about that too. But these filtration slits are made by the protoplasmic extensions of your podocytes. When they're coming together like this, it creates a gap or space, okay, of these pedicels or end-fit, okay, and those are called filtration slits. Okay, we'll talk. And I just said this whole thing as a filtration slits. Okay, now after that, we also have to talk about one more thing. Is what is that thing? Is that, you know, these guys right here, what is the unique part of these guys is that these protoplasmic extensions or pedicels are held by different types of proteins, okay? There are many proteins, but I'm going to mention a few of those. One protein is protein, this one protein. This protein is coming together right here like this, okay? And it is also creating a gap, this protein. Do you know what is the name of the protein is called? This protein is called Nephron. Okay? And I'll talk about what is the role of that. And what is this regulating this protein is, there's something called right here. Okay? This is kind of sort of like regulating this proteins, and that rate is going to be my podocin. Okay? And then there's also one more thing he quickly mentions about that too. Okay? I'll just make it this one. And this is called my alpha actinin 4. Okay? There are other ones too, but we'll just mention this 3 of those. So, see, this nephrin, this a protein, it is actually branching out from the food process, or this protoplasmic extensions of these podocytes, okay, and then it is coming and it is also creating as a barrier, okay. This is nephrin, okay, and then these nephrins proteins are regulated by this proteins called podocins, and this is also, there is also alpha actinin 4, which sort of like maintains the cytoskeletons of the cells, okay. Now this podocins, this nephrin creates sort of like a barrier, okay. It doesn't allow a lot of things to get into. Usually it's a diameters of like 7 to like 9 or 10 nanometers of diameters of the nephrin, okay. If there is a mutation in his gene, like for, I think the name of the gene is, it's like NPHS1 and this one is like NPHS2, okay. These are genes, okay. If there's a mutation in the nephrin gene, you know what you can get? You can get some kind of nephrotic syndrome. For instance, you can get something called congenital, okay? Congenital nephrotic syndromes, and this is sort of like a finished type, they call this finished type of congenital nephrotic syndrome, okay? Or glomerulonephritis, that's what they call this, okay? And there is a podocin, okay? And if there's a mutation, the podocins are the genes on this, then you can get some conditions called the steroid resistance, okay? Glomerulonephritis, okay? And we'll talk about a pathological condition too. And then if you get this alpha actinin 4, then you can get something called focal segmental glomerulosclerosis, okay? It's called focal, okay, segmental, okay, glomerulosclerosis. It's a nephrotic syndrome, okay? This can give congenital nephrotic syndrome, it's called finished type, and then this is called, as I said, steroid resistance, okay, the glomerulonephrotic syndromes. So these are different pathological conditions, okay? If there's a mutation in the genes, okay? That's the reason why I quickly mentioned about this, okay? These are very, very important to know about this, okay? Now, so as we talked about, in order for the filtration to occur, it has to pass all those barriers in order to come to this area right here, okay? And then to go to the proximal convolutive itself. Because it has to pass this 3 different barriers, we call them as ultrafiltrate or glomerulofiltrate, okay? Whatever product that comes in down here, we call them a glomerulofiltrate or we call them ultrafiltrate, okay? And this whole area, okay? This whole area, whenever we talk about the glomerulus, endothelial cells, basal membranes, and we talk about this podocytes and all that, then we're talking about the Malpighian corpuscles or renal corpuscles, because renal corpuscles includes, or Malpighian corpuscles includes your glomerular capillaries and also the Bowman capsules, okay? That's what we're talking about. And these 3 things, okay, it has to passes for the for filtration to occur, okay? So these are just the structures that I wanted to mention about it, okay? So whenever we talk about the glomerulofiltration rate, okay, these are the fundamental we have to use to have a greater understanding of the glomerulofiltrations, okay? And all the things that I mentioned to you guys, you know, these are very important, okay? And then we talked about the anatomy. I tried to get a little bit of histology too, and we also talked about the structures of the kidney, okay?